So welcome. My name is Brian Schneedy, and I'm the Outreach and Engagement Manager with the Dutchess Land Conservancy. I want to thank you for joining us on our fourth webinar in this season of our Earth Matters webinar series. We honestly enjoy hosting these events, get a ton out of them ourselves, and are always happy to see so many guests. Tonight, we are very excited to welcome the Cary Institute of Ecosystem Studies' own Dr. Jane Lucas, as she gives her presentation on soil health in the age of antibiotics. But first, we got to go over some basic logistics about tonight's webinar, followed by a brief introduction to the Dutchess Land Conservancy along with tonight's presenter. First, you are muted to ensure that there is no interruption during the presentation. Second, the raise hand function is disabled. If you have questions, you can enter them into the Q&A box at any point during the presentation. You can access this box at the bottom center of your screen. These questions will be addressed when the presentation is done, or by our team during the presentation if possible. And if there are a great deal of questions, we'll try to combine them toward the end. Third, the chat is disabled for this event. Fourth, closed captioning is enabled for this event. To turn this function on, please select the CC icon located at the bottom of your screen. And finally, I'm going to hand it over to Julie Hart, our Education Director, who will tell you a bit about the DLC, this webinar series, and our guest presenter. Julie.
Thank you, Brian. Good evening, everyone, and thank you for joining us. I'm Julie Hart, an ecologist and educator here at the Dutchess Land Conservancy. Many of you are probably familiar with the DLC, but for anyone who's not, let me give you a quick overview. Dutchess Land Conservancy is a private nonprofit land conservation organization located in the Hudson Valley of New York State, and we're dedicated to preserving the farms, forests, wetlands and waterways, open spaces, and wildlife habitats of Dutchess County. We are an accredited land trust, and since our founding in 1985, we've worked with hundreds of landowners and protected over 46,000 acres of land. Many thanks to the Molly B. Schaefer Education Fund for sponsoring this event. We are so excited about our new season of Earth Matters, the DLC's winter webinar series. We're bringing you interesting speakers on the first Tuesday of each month through April, and this season we are digging into the topic of soil health. We're learning about such topics as soil food webs, regenerative soil management practices, composting, and the ecology of soil health. Our theme is very simple. Soil is not dirt. So let's get started. Tonight, we are thrilled to welcome Dr. Jane Lucas of the Cary Institute of Ecosystem Studies, and that's an institution that's just down the road from our DLC office here in Millbrook. And full disclosure, I used to work there, and so nutrient cycling and soil health is a topic very dear to my heart. Jane received her PhD in ecology and evolutionary biology at the University of Oklahoma, where she studied how nutrient availability and antibiotics influence tropical soil communities. Jane's work examines how livestock antibiotics affect soil microbes, soil carbon storage, nitrogen cycling, and the rise of antibiotic resistance. By investigating the linkages between agriculture, the environment, and public health, and working with farmers and government agencies, Jane's work serves to encourage sustainable agricultural practices that support healthy ecosystems and meet global food demands. So with that, I will turn it over to Dr. Lucas, who will talk about soil health in the age of antibiotics.
Wonderful. Thank you all so much. It's such a pleasure to get to talk to you all this evening. I am just always excited to talk about some of the work my lab is doing as well as others. And tonight, I'm going to focus a lot on soil health and antibiotics. And I think that oftentimes we don't think about these two concepts at the same time, although of course I spend quite a bit of time thinking about them together. But hopefully by the end of this talk, you'll understand why antibiotics and soil environments are so tightly linked. And it's important that we understand how they're connected to really understand soil and soil health. So to begin with, I want us to go back in time about 100 years and imagine that we are in London, 1928. It's a complicated time. We're coming just to the end of the Roaring 20s, where there's been quite a bit of progression. In fact, in 1928, women have just officially been allowed to vote, and as long as they're older than the age of 21 in London. So they're seeing some progress, but we're also seeing big labor problems arising, as well as a big catastrophic flooding of the River Thames. So it's a complicated but exciting time to be in London. And in particular, there's something that is pretty big that's about to happen. And that is really a story of a young scientist named Alexander Fleming. He's working at a hospital called St. Mary's Hospital in London. And what Alexander Fleming is doing is he is really interested in studying infectious bacteria, the types of things that are causing diseases throughout the world. Now, like many microbiologists, what Alexander Fleming does is he captures bacteria and he attempts to have it grow in a laboratory environment so he can understand what allows it to do well, proliferate, and what might limit or stop infections. And so many of you might have seen this kind of typical photo of how microbiologists grow microbes. And one way we do this is by plating them. And so we take this nutrient-rich broth, which is really kind of like a jello substance, and it has all the things that many microbes need, and it allows microbes to proliferate. And so it's likely that Alexander Fleming was growing these bacterial colonies in his laboratory. But like many of us, he felt like he was a bit overworked, and so he decided that he was going to take a vacation. So he kind of ships himself, he takes a train out to the countryside, and for 2 weeks, he leaves his bacterial colonies alone in the lab. And while he's out of the lab, some mold spores kind of lift up and land on some of the bacterial plates. And then what happens is what you can somewhat see here is similar to these discs. The mold created these zones of inhibition. It stopped the bacteria from growing. And so when Alexander Fleming returned, he noticed that in these plates that had mold spores on them, there was this inhibition of bacteria. And this is really credited with kind of the discovery of antibiotic compounds, because Alexander Fleming took that time to say, wow, there must be something going on with this mold that's limiting the ability of microbes, and maybe we could capture this. Now, I do want to say a brief aside. While we kind of credit Alexander Fleming for discovering what we think of as antibiotics now, plenty of people, in fact, as early as the 1700s, had kind of come up with potential ideas that chemicals could stop bacterial growth. But in general, we kind of credit Alexander as the modern founder of antibiotics. And I think it's also kind of amazing to know that it's only been since 1928 that we've understood that these compounds exist. So in just 100 years that these, world-changing drugs have changed our world. So from the study of Alexander Fleming, there's a couple things that I want to emphasize that occurred. Well, first, and probably most importantly, we discovered really this concept of antibiotics. We also noticed that it's really important to take a vacation, because if Alexander Fleming hadn't left for a while, it's very likely that he would have seen those mold spores land on those bacterial plates and thrown them out and thought, oh, these are ruined, I can't use them anymore. But it took moving away and allowing nature to run its course for a couple weeks in order for this discovery to come about. And then finally, and I think this is such a wonderful and great part of his story, is that it really emphasizes how important it is to sometimes move slowly through science and to be observant, and that sometimes our biggest discoveries are not where we were trying to get to an end goal, but in fact come by taking that time. And I credit Alexander Fleming because I think he had some really, strong hubris. He understood that, it was his observation that was such a great discovery. In fact, this quote from him says that penicillin, which is the compound he kind of discovered, started as a chance observation. And his only merit is that I did not neglect this observation. And I think that's such a lovely and hopefully, helpful motto to take with life. Now, since his discovery back in 1928, the kind of world of antibiotic development has exploded. In fact, many people credit the discovery of antibiotics as one of, if not the most world-changing discoveries in the 1900s. And clearly it's had a huge impact on human health, and we use them in a wide variety of human-based systems. But I want to remind you all that while we have known about antibiotics for about 100 years, nature has known about them and has been using them for millions and millions of years. And I want to tell you just a couple stories of how nature has been able to use and capture the power of antibiotics in their own stories. So recently, in fact, just a couple years ago, scientists found a whole new class of antibiotics living in the roots of wild bean plants. So if you don't know a ton about beans, many of them have these important relationships with symbiotic bacteria, normally in the genus Rhizobium. And what these Rhizobium do is, you can see here in this blown up part of the photo, is that these bacteria colonize the roots and they create these little balls that are called root nodules. And what these bacteria are doing is they're coming in and they're fixing nitrogen and supplying it to the plant. So here you can see on the left, this is an uninoculated, a plant that doesn't have those root nodules. And here's a nice healthy plant that does. So these bacteria provide a really great nutrient to the plant. But what scientists found is that they don't just provide nutrients, but these bacteria are also capable of producing an antibiotic that actually keeps negative, bad, other types of bacteria out of the plant roots. So they're helping the plant by giving it nutrients while also defending it against pathogens. And I think this is such an incredible new world, and who knows how many new antibiotics are out there living in root systems that we've yet to discover. Now, I think this is a relatively new world, but maybe you all have heard about a really classic case of nature using antibiotics, and that's with the wonderfully charismatic leafcutter ants. So leafcutter ants are kind of those, quintessential first shot of a nature documentary ants that you see. They are climbing up into the forest canopies and they're clipping little bits of fresh leaves. And now these ants aren't actually eating the leaves themselves, but instead they bring the leaves back to their nest and they feed it to this fungus. And this fungus is actually what feeds and is acting as a food source for the colony. And so there's this really tight relationship where ants need that fungus to grow, but the fungus needs them to bring fresh leaves in order to grow itself. But you might imagine that what helps some fungus grow can help a lot of different types of microbes. So the ants have to work really hard to make sure that their beneficial, their good fungal colony doesn't get overtaken by other negative microbes. And so what they actually have developed are these amazing pockets inside of their body that have bacteria in them. And then that bacteria can secrete antibiotic compounds that the ants rub all over this beneficial fungus, and it helps keep that fungus healthy, happy, and have non-pathogens come into the system. So it's this really cool three-way interaction where the ants are feeding the fungus and they're keeping the fungus clean, and the fungus is helping keep the ant colonies clean. And I'll say, just as a side note, I started my work in ecology in an ant lab, so I'm always fascinated and have plenty of stories about ants, and always happy to talk about ants if people have more questions about them. But of course, as you'd imagine from the topic of this series, what we really want to focus on is the link between antibiotics and soil environments. And that's because soils have been an amazing source for antibiotics for almost all the antibiotics that we've found. So in fact, here is a list of many common classes of antibiotics. Maybe some of you have taken them yourself, things like penicillin, streptomycin, tetracycline. All of these potent drugs have been found being produced in the soil environment. And I would say that, these are just some that we've discovered. There's probably many more out there. We just haven't discovered them yet. But why? Why are soils such a hot spot for antibiotic discovery and production? Well, the big reason for this is that soils are incredibly diverse. They have a huge variety of microbial life living inside of them. In fact, if you went out and just took a tablespoon of soil, you would likely find more microbes in that tablespoon than there are humans on the planet Earth. So it's an incredibly diverse environment. But when you live in such a diverse environment with all these microbes, you also end up having to compete with them because nutrients and space is limited. So what microbes have done is they've developed defensive compounds, which are antibiotics. And that's one way that they can fight off competitors and kind of take over a resource. So really, the soil is such a hot spot for diversity of microbes and also this competitive dynamic. So it's a great place to look and find antibiotics. So ultimately, what I want you to really kind of grasp first and foremost is that antibiotic compounds are naturally occurring compounds. You know, we so often first think of them in their medicinal context in this very sterile sense, but in reality, they are such a natural part of our ecosystem. And in fact, if you went back into your yard or out into a nearby park, odds are very high that you'd be able to find antibiotic producing bacteria in your soils. And I think this world of natural production is so fascinating. You know, we know that there is the potential for antibiotic production in soils, but there are so many more questions out there, like how many different types of antibiotics, when is it being produced, for how long do they produce it, and why? So lots of really fascinating natural questions. But we as humans are changing this natural distribution of antibiotics. And we're doing this primarily because we are using antibiotics pretty frequently in our lives. Now, one of the biggest ways that antibiotics are introduced into the environment through human pathways is through wastewater treatment plants. And so many of you might know, if you're familiar with the Cary, some of the wonderful work done by my colleague, Emma Rosie, and she has really looked at how pharmaceuticals that come off of wastewater treatment plants, and in particular antibiotics, move through our waterways and the effects these can have on all the organisms and nutrient cycling processes happening in our water systems. So if you're interested in that, please look her up because she's got some really fascinating work. But another way that antibiotics are entering into our ecosystem is through livestock production. And today, I'm going to focus a lot on the livestock portion of antibiotic introductions, because this is really where my lab has been focusing for quite some time. And I also want to focus on it because livestock production is something happening all over the earth. And it has kind of different impacts in different environments. And so I think it's just this wonderful natural, world is a natural ecosystem and a natural experiment that's allowed us to ask many interesting questions. But another really big reason we have looked at antibiotics in livestock is that that's where most of the antibiotics we're producing are going. In fact, about 80% of all of the antibiotics that we produce, the most recent estimate is about 14 million kilograms of antibiotics every year are going into livestock industries. And there are a couple of reasons for this. You know, the first might be quite obvious to all, which is, of course, if an animal gets sick, we want to be able to treat and help them get better. But we also often use these compounds to prevent disease. So many times we'll use small amounts of antibiotics to prevent disease from ever outbreaking within a system. And especially in areas where animals are really tightly packed and congregated, you can imagine that disease could spread fast. So keeping that disease out is really important. Another big reason, though, that we've used antibiotic compounds is that they have caused this interesting artifact of increasing the yield, the size of animals, when we give them these kind of low subtherapeutic levels of antibiotics. Now, I'll say that this is a really fascinating line of inquiry, and we actually don't have 100% clarity on why antibiotics cause animals to grow bigger. But it's true that between, they can grow just a couple percentages to almost 40% bigger with these antibiotic compounds. So you can imagine for producers who are trying to maximize their profit and already living on maybe, kind of marginal gains, being able to increase your yield is really important. So there's a big demand to use these compounds. But I want to remind you about something that maybe you don't think about every day when we take antibiotics. So when humans or animals take a course of antibiotics, our body is not able to completely digest those compounds. It's not like we take a pill and that whole thing dissolves in our stomach and gets used. In fact, a lot of times, a portion of the dose comes in one end and then comes out the other. And so this is true in humans, and it's also quite true in animals. So here is a graph of the excretion rates from cattle of common classes of antibiotic compounds. And what I want to emphasize here is, again, that, just a couple percentages of the dose to almost 100% of the antibiotic can be eliminated through kind of urine or manure because of how inefficient we are at breaking down some of these compounds. I also want to emphasize that, oftentimes it's the exact same compound coming in one end and leaving the other, while other times it can be a slightly metabolized version, but it still might be active and able to affect microbial communities. And if we think about the fact that our lovely little dairy cow is producing an insane amount, about 82 pounds each of manure every single day, I feel like that's a fact that just always blows my mind. But if this waste is from an animal that's getting these continuous antibiotic courses, you can see how we're creating a continuous input of antibiotics in one end, out the other, and into the ecosystem. And so this kind of input is really a central focus of my work as well as some of my colleagues. And we really want to understand, if these are entering into the environment, as we know they are, what effects are they having on soils? And do we need to do some different types of practices to make sure that we're maximizing our soil health while also helping benefit producers and human health? So today I'm going to walk you through kind of 3 topics. The first and most major component of my talk will be looking at the work of how livestock-derived antibiotics are entering into our soils and the effects they have on our soil environment. Now, I also want to talk about antibiotic resistance because I think, it's very natural that when you talk about antibiotics, you also want to talk about resistance because they are very tightly linked. And then finally, I want to just talk about some of the next steps of where our work is going and also give you all some tips about how to kind of properly manage antibiotics in your environments. All right, so let's start first with understanding antibiotics in the soil environment. So my lab has been doing quite a bit of work exploring this question of how livestock-derived antibiotics come into the environment and the effects they're having. And a major way that we do this is that we go to our kind of experimental farms and we give a normal course of antibiotics to some cows, and we take other cows and keep them as our controls. They're not given any pharmaceuticals. And then we have this very glamorous job where we have to stand behind the cows and wait for them to do their natural business. It's really, truly such a glamorous experience. But that allows us to collect the manure and waste from these animals. And that way we have manure that's from treated animals as well as manure from untreated animals. And we have quite a few different types of compounds. Here's a list of some of the common ones our lab has explored. Things like penicillin, streptomycin, tetracycline. These are things you probably have heard of before. But I also want to emphasize that we spend a lot of our time focused on monensin, which is a class of antibiotics that I'm going to guess many of you are not familiar with. And that's because this class of antibiotics is only used in livestock systems, but it's one of the most frequently used antibiotics. We actually say that, general policy is that monensin is a better choice because we don't use it on human health. So the, kind of threat of resistant spillover is lower. And so once we have this manure from our different cattle, we bring it out into the field and we ask some important and somewhat, clear, simple questions. The first thing we often ask is, do, antibiotics introduced to cattle through the, natural livestock system change the microbiome? So what microbes are present in our soils? Now, we also know that soils are not just full of microbes. In fact, they're teeming with tons of diversity. And so another natural question we ask is, what are the effects that these different compounds may have on the invertebrate community? And then finally, if we're seeing these changes in the communities in our soil, are we also seeing changes in the bigger ecosystem processes that microbes and invertebrates control? Things like nutrient cycling or decomposition. So I want to walk you through some of the results that our work is highlighting and give you kind of an overview of some of the general trends that typically emerge from our work. Now, the first result we see here is probably one that you'd assume would be the case. And that's that when antibiotics are entering into our soils, it decreases the amount of active microbes in the soil. So here on the left, you can see we have our control soils that have not been experiencing any antibiotic course. And then we have soils that have low or high doses of antibiotics coming into them. And in general, we see about a 25 to 50% decrease in active microbes. And that's maybe what you'd expect. Antibiotics are supposed to kill microbes, and that's what we're seeing. What we also see is that the microbiome of the manure and then the soil underneath that manure is different if it came from cows that were given an antibiotic course compared to cows that were not given an antibiotic course. And so this is not just the number of microbes present, but the types of microbes that are present are different. And again, I think this can kind of be somewhat intuitive because antibiotics are likely not going to kill every single microbe in the environment, but it'll help some microbes and harm others. And that causes the composition, what microbes are present in our soil microbiome, to change. And interestingly, we also see that when we have antibiotics entering into the environment, fungal communities are generally tending to dominate those ecosystems. And I think this is a particularly fascinating result because it really kind of makes you think about what the bigger consequences are for these introductions. Now, I think fungi are doing better in the environment because normally they have to compete with lots of bacteria to gain resources, including nutrients and space. But most of the antibiotic compounds that we use in livestock, as well as our own health, are antibacterial. So you can imagine that if antibacterials come into the environment, they knock down some of the competition. And so fungi are able to kind of exploit all of these new resources and take over. And I think this is particularly interesting because we generally think that soils that have more fungal growth in them are healthier. Whether or not that's entirely the case is definitely still up for debate and really depends on what you're trying to get those soils to do. But I do think what this emphasizes is that the antibiotic response is really complicated. It's killing some microbes, but it's helping others. And whether or not that's good or bad is really kind of a matter of what you want your soils to do. Now, as I mentioned, soils are also filled with many more organisms than just bacteria and fungi. And I'm guessing that many of you were able to log into the wonderful talk earlier in December about soil food webs. But if not, I just want to remind you that there are all these really fascinating webs happening below ground. And normally what happens is we get this decaying matter that comes in, and initially the bacteria and fungi colonize it and break it down. And then there are nematodes and mites and columbella that consume those microbes. And then they're consumed by other types of invertebrates. So the soil food web is really a fascinating component of our soil health. And we really want to make sure that we're keeping a healthy soil food web, because that really affects the quality and the nutrient content in our soils. But naturally, it makes us want to know, we know soil food webs are important. So what effect might antibiotics have on soil invertebrates? So in those same soils that we take from a antibiotic treated manure system versus an unantibiotic treated manure system, we extract the microbes and we extract all of the invertebrates in them. And the way we do this is through a really somewhat simple but elegant process called Berlese or Tolgren funnels. And I wanted to just walk through this because if you or anyone is interested in soil and want to understand more of the diversity, this is an easy technique that you can actually do at your own house. And what we do to look at all the invertebrates in our soil is we take our soil and we put it in a funnel. Now, we have specific funnels for this, but you could actually take just a 2-liter pop bottle and turn it upside down, cut it in half, turn it upside down, and there you go. You have a funnel right there. And then what we do is we put a cup with alcohol underneath it. And then we just put a lamp, which acts as a heat source above it. So most soil invertebrates don't like it to be too hot. So what they do is they move away from that heat and they run down the funnel. And they drop, and I know it's a little morbid, but they drop into our alcohol collection, and there we have them preserved and can actually look at them. And so I know it's a little bit fuzzy, but this is very much something that you might see if you extracted invertebrates from your own soils. So here we have different mites and columbella, those things that sometimes you see on top of snow, the snow fleas, they're all over our soils. We have different types of mites in our ecosystems, and it's really fascinating. In fact, the first time I looked at the results of a Berlese funnel, it changed the way I thought about soil, because this could be just, a couple 100 grams, not a lot of soil, and it could have tons of this amazing diversity. So back to our study, we know that these invertebrates are important, and what effect do antibiotics have on them? Well, we see some really interesting results. We see that microbivores, so invertebrates that really their main food source is microbes, actually do a lot worse when antibiotics are in our ecosystem. And again, we see that even with a little bit of antibiotics. We don't need a high dose. We can see it with a low or high dose. And this kind of makes sense, right? We're bringing in antibiotics, we're probably killing a lot of the microbes. And so those individuals that need to survive on the microbes have less food, they're decreasing it. Though it's also quite likely that the antibiotics themselves are directly harming them. And that's a line of inquiry we're kind of exploring right now. But interestingly, we see that predatory invertebrates, things that feed on other invertebrates, actually do better when we have antibiotics in the environment. And in fact, more antibiotics helps them do even better. So these might be things like spiders or predatory mites. And I think possibly the reason that they do better is that all of a sudden, when we bring antibiotics in, it's killed a bunch of those microbivores. And so it's kind of a feeding frenzy that allows predators to do better. But I'm really curious to see, most of our studies are over just a couple weeks to a couple months, and whether or not predators would do well long-term is definitely a question we have. So we see that kind of what's present in the community is changing, but we also see, and this gets a little bit, like nesting doll, but the microbes that live inside of insects are also changing depending on their type of diet. So some of the work we've done has focused on dung beetles. And when we give dung beetles manure from antibiotic treated cattle compared to untreated cattle, we see that their microbiome changes. And I think this is important to know because invertebrates in particular can be really reliant on a healthy gut microbiome because that's where they're getting most of their nutrients from. They need those microbes to grow. And if we're changing that microbiome, we might be changing, how well they're able to survive and possibly causing some long-term consequences for dung beetle populations. So just to summarize from those antibiotic effects, we see that when you add antibiotics to cattle, it changes the microbiome of their manure, and it also decreases the number of active microbes. Similarly, we see that the soil underneath these manure pads is different in those antibiotic environments. We have more fungi, but less overall active biomass, and the composition is quite different. We also see some effects on the soil invertebrates. We see that who's present changes, as well as decreases in some types of invertebrates and increases in others. And finally, we see that the microbes inside those invertebrates are changing. So we're seeing this kind of cascade of effect. And in particular, our study is emphasizing that antibiotics are changing our soil food webs. And, soil food webs are so important. They're such a critical part of healthy soils. And so it's really important we know how and when and how severely they're changing, because these potentially could have really big impacts on how we manage our soil. But I do want to emphasize that, to some extent, if we aren't studying those effects, maybe it doesn't matter. So naturally, a big follow-up question our lab is exploring is, so we see these changes in who's present in the soil, but does that actually change what the soil is doing, the function of these soils? And the answer is yes, we are seeing some big shifts. One really interesting response we've seen in some of our studies is that when antibiotics come in, it actually increases the bioavailable nitrogen in the ecosystem. Now, I think a big reason for this is because we're killing lots of microbes, we're opening them up and kind of causing this flood of bioavailable nitrogen in our systems. And so maybe, at least in the short term, this could be beneficial because it acts as almost a natural fertilization. We also know that some antibiotics actually have nitrogen in them, so that could also be driving some of those nitrogen responses. We also see that antibiotics can cause an increase in the pH of soils. And for those of you who are farmers or, gardeners or just interested in soil health, you probably know that pH is a really important indicator of how healthy your soils are. And most soils really like to live in that neutral range. So when we're changing the soils and increasing them and pushing them, above 7, 8 pH, that's going to cause big effects on how they function and what microbes can continue to live there. So it's important we know how that pH is changing because it might influence how we manage our soils. And additionally, we find that oftentimes antibiotics have led to a decrease in the water holding capacity of soils. Now, this is a term that's very much what it sounds like. Water holding capacity is how well soils hold onto water, as it rains. And whether or not they can hold water is a really good indicator of soil health. And now most of us want more water holding capacity because that means that we don't need to be irrigating, we don't need to add water because they can actually hold on to it for a while. And this is really important in drought kind of susceptible areas because increased water holding capacity means you might be able to buffer against periods of, prolonged drought compared to decreased water holding capacity. So we see that there's some big effects on the, characteristics of soil. But my lab is also really interested in the influence antibiotics have on things like greenhouse gas emissions. And so I just want to remind you that maybe you don't think about this too often, but soils are just like us. They are respiring CO2 all the time. And in fact, it's normal. You should be having soils respire CO2 because microbes like us are breaking down materials. And as they do that, they breathe out CO2. But we want to make sure that they're breathing out the correct amount of CO2 and not too much where they're actually losing carbon. So one of the big questions we consistently ask in my lab are, how are antibiotics affecting CO2 production in our soils? And so from one of the studies that we did, again, looking at those low or high doses of antibiotics, we found this interesting result, which is that the control manure from an untreated cattle had no difference from our low or high antibiotic treatments. So this means that the amount of CO2 coming off of these soils is the exact same, which makes it seem like, oh, well, I guess antibiotics don't affect respiration. But I want to remind you that I told you earlier, our soils have far less active microbes in them. In fact, about 25 to 50% less active microbes, but they're still producing the same amount of CO2. And so what that means is that antibiotics are coming into the soil and increasing the per unit, the mass specific respiration of our microbes. And so just to maybe put that into context, I want you to think about a track. And imagine I asked you that you need to go around a track one time. Now you can walk around the track or you can run around the track, but in the end, you're going to do the same amount of distance. Now, if you run around the track, you're going to burn more calories. And in the end, you're probably going to be working harder, breathing a lot more. And so you had to do more work to do the same job as somebody who is just slowly walking around the track. Well, that's kind of what antibiotics are making our microbes do. It's making them work harder to do the same job. And this is a problem because that means our microbes are less efficient. They're kind of producing more CO2 to grow. And that means that less CO2 is likely sticking around in our soils. And that can be a really big issue because soils are our main terrestrial carbon store. So we want to keep carbon in our soils. We don't want it to be leaving more and more. So from this work, we can see that antibiotics can come in through livestock practices, and they can disrupt and change our soil food webs, as well as the function of our soils. But again, it's a complicated result. Sometimes these effects may be really severely negative. Sometimes they're just a little bit negative. Sometimes they're neutral, and maybe even sometimes they can be beneficial. It's a complicated relationship. And to add even more complication to it, the antibiotics effects can be really variable depending on the type of antibiotic we use. Now, I've shown you kind of some summarized data, but I also want to say that on a finer scale, we see that some antibiotics can have much stronger responses in the soils and their function than others. And something actually like penicillin, which we started our story with, has pretty low effects on our soil environment. But something like monensin has potentially really big impacts. We see that they really dramatically change the soil microbiome and cause a much less efficient soil community. And another reason I want to emphasize that is, while I think it's really important that we are thinking about smart antibiotic use, we've really encouraged monensin use because that's what we don't use in human systems. But it is one of the more disruptive antibiotics on our soil system. So it's really important to me that when we're making decisions about what types of antibiotics to use and when to use them, we really consider the One Health perspective, which means we're considering human health, we're considering animal health, and we're thinking ecosystem health, because in reality, all of those are tightly linked, and we want to make sure that we're making decisions that kind of minimize harm and maximize benefit. And so I think there are tons more questions out there. We're really just beginning to scratch the surface of how livestock-derived antibiotics are affecting our soils. And I wanted to just briefly highlight a study that we just started recently called Stressed Out Soils. And this is examining how multiple global change factors are affecting soil and human health. So if you're ever on the Cary campus, you might see our plots up by our weather station. But what we're doing in this study is we're adding different antibiotics to the soil, but we're also increasing the temperature and changing the amount of precipitation, because this is what's going to happen in soils around the world. We're going to be seeing warming and drought happening at the same time that antibiotics are entering. And from this, we really want to know, how far can we push our soils? Are they, able to function with lots of these inputs, or is there kind of a tipping point? We also want to know, can we look at the soil and say, this soil is stressed, we need to take care of it? Or is it really hard to tell? And then finally, because I mentioned, we want to look at the human health link too, we're really interested in how these different stressors are affecting ARGs, or antibiotic resistant genes. And so for the last bit of this talk here, I want to take a little bit of time to talk about antibiotic resistance in the environment. And a big reason for this is that it's hard not to talk about antibiotics and also talk about antibiotic resistance, because they are so tightly linked. And of course, as much as antibiotics are a naturally occurring compound, so too is antibiotic resistance. And so I want to emphasize that, just because antibiotic resistance might be present, it doesn't mean that it's this major threat. It's just when it goes to a balance that's kind of unhealthy that we need to be concerned. But I also want to emphasize that antibiotic resistance can come into the environment really quickly. So I just want to take a second to show you a really cool video done by a group at Harvard. And so let me just start this video. I want to make sure it all works. All right, so what you're going to see here is called the mega plate Petri dish. And it's this large, oh no, I hope that I didn't. Okay, great. So what you'll see here is this dish that they've grown bacteria on. And they start on the ends here with no antibiotics in these environments. And then they slowly increase these concentrations to be 1 times, 10 times, 100 times, and 1,000 times. So as the microbes are growing across this plate, you'll see that they start over here on the ends, and then they grow towards the center. And what they wanted to see is how quickly does E. coli adapt to antibiotics and become resistant. So here you can see, oh, did it stop? Oh, no, it's still moving. You can see that the microbes grew, but they hit that first line, and all of a sudden some colonies are able to adapt and become resistant. And so slowly they're going to continue forward, but you can see, especially on this side of our screen, that it came up to that 10x and just kind of flew past it. So these microbes initially were not resistant, but really quickly have been able to adapt and grow and be able to comfortably live in a high antibiotic environment. And remember, at the end here, this is 1,000 times. This is a really potent antibiotic environment. And these microbes are able to grow in this ecosystem. And you'll see they're going to overlay. This is a tree of those different microbes. And what's crazy to me is that all of this happened in less than 2 weeks. So these microbial communities were able to grow in such a short period of time. And so what I want to emphasize is that, yes, antibiotic resistance is a naturally occurring concept, but our misuse and overuse of antibiotic compounds and introduction of them into the environment is accelerating how quickly this resistance can occur. And of course, it can happen really fast, right? So we need to be really smart about our antibiotic use to make sure that we aren't spreading really rapid rates of resistance. I also want to emphasize that the World Health Organization really emphasizes that antibiotic resistance is one of the most important, largest threats to global health, food security, and development. So this is a big problem that we need to understand. And in particular, I think it's a really big problem because antibiotic resistance can affect anyone. You don't need to be immunocompromised or living just in a specific area. It happens all over the world. And so we know that antibiotic resistance is such a big thing. It's a big concept that we need to understand. And we know that it happens naturally. But I'm always shocked at how little we know about antibiotic resistance in the environment. And in fact, if you just ask someone how much resistance is outside my door, the answer would be, I don't know. And so a really big question my lab has been tackling is, how does antibiotic resistance differ across different ecosystems? And what characteristics, what is it about the environment that might increase the amount of resistance in the ecosystem? So for this, we've gone to 220 sites across the United States so far. And what you'll see is that some of our sites are uncultivated. Those are the blue dots. So these would be places like a native forest or grasslands, areas that don't have a lot of human influence. And then we also collected soils from cultivated sites. So things like croplands, pasture lands, as well as people's lawns. And we looked at all of the antibiotic resistance in these sites. And one of the very basic but important questions we've asked is, how does antibiotic resistance vary biogeographically? So I'm going to show you some of our initial results. The first thing we see is that the number of different types of resistance can differ quite a bit depending on where we are. So you can see maybe on the right up in the Vermont area, we have kind of high rates in our cultivated lands of antibiotic richness. The height of these sticks represents how much richness is there. But then you can see maybe in kind of the southern Colorado that these are areas with less antibiotic richness or resistance richness. We also can see that not just the types of resistance, but how much resistance, how many genes are present in the soils can really vary dramatically. And again, kind of some of our spots in the Northeast area generally tend to have some pretty high rates of antibiotic resistance compared to areas in kind of that southern, Southwest, Utah, Colorado area generally have lower rates of antibiotic resistance. And I want to emphasize, this is just the first study. We definitely have lots more to explore, but this is one of the first kind of comprehensive maps, and we're still missing plenty of states, so it's not that comprehensive, to really show this natural variability of environmental antibiotic resistance. But some interesting trends are emerging, and one that's really jumps out is that in general, cultivated lands, so areas where humans have a lot of influence, are increasing in antibiotic resistance. In fact, about 47% more antibiotic resistance in those environments compared to our what you might consider natural sites. And here we kind of break it down by the different types of environments. And one pattern that I just want to highlight is that, somewhat surprisingly, human lawns generally had the highest rate of antibiotic resistance. Now, again, I don't want to say, sound the alarm, don't go outside, be aware of your, don't be afraid, but it is important that we know that these areas that humans come into a lot of contact with also might be areas that have hot spots or high levels of antibiotic resistance. And, you don't have to take in this whole graph. It's kind of a crazy way that we demonstrate all this. But what I also want to say is, yes, the numbers of resistance differ, but also the types of resistance. And so something like our dryland forest has a lot more tetracycline resistance than some of our other areas. And these are really interesting patterns that we're trying to dive into further. But in general, what our results highlight is that different types of environments have different rates of resistance. And this is an important first step for us to understand how we're going to handle resistance in our soils, and also potentially areas we need to be more concerned about. But I do want to bring it back a little bit to the livestock presence, because we've done a little bit more detailed work at how the presence of livestock are affecting resistance across our different sites. And so in a study, we went out to some of our sites across the United States, and we specifically looked at antibiotic resistance in soils where the livestock were highly congregated in areas that have lots of manure inputs. And then we collected right next to it what we call a reference, or kind of a control soil. And we looked at how much antibiotic resistance were in these different soils. And so here you can see all the dots of places we've gone to, and the size of the dots represent how many antibiotic resistant genes, how many are present in the soil. And these blue dots are our reference sites. And as you can see, they're not that big because they didn't have a ton of antibiotic resistance in them. But when we looked at the areas where cattle congregates, we saw that there were much higher rates of antibiotic resistance. But I also want to emphasize that this was variable. You know, actually up here in our New York site, the rates were really comparable. There weren't really high rates, but in our more southern area, we did see quite a large increase of antibiotic resistance where cattle congregate. And it wasn't that all types were increasing, but we do see that consistently that manure input was driving some of those resistant gene abundances up. So from this work, we can see that antibiotic resistance varies across environments, but people's lawns and areas where livestock congregate generally tend to be some of the most highly abundant antibiotic resistant environments. And so this, I think, is just the first step, but we have lots and lots of remaining questions. So we're excited to keep diving into this. You know, one of the big things we want to know is, how long does this resistance stay in the environment? It's this really dynamic concept. So we want to continue to explore that. We also want to know if there are certain practices, you know, fertilizing or tilling, all these different ways that we manipulate our soils that maybe increase or decrease antimicrobial resistance. And I do want to emphasize, I forgot to put this slide in, but I think it's so cool. I recently was talking with a colleague, and they actually are finding that when you have lots of earthworms in your soils, they're actually able to eliminate some of the resistance by processing that soil. So there are things that can eliminate antimicrobial resistance from our soils. And then another, you know, really important question we have is, particularly on livestock, is it the manure or is it the antibiotics or both that's driving these antimicrobial resistance increases? And so we're continuing to work on this. I also want to shout out a great project that I get to be a part of with the University of Connecticut, where we're looking at antimicrobials and resistance that's both in the soils and how it's moving into that aquatic system and how far down our aquatic systems those effects are occurring. So, you know, we do see these environmental effects, but you know, does this actually mean anything for human health? And I do, again, just really want to emphasize that my goal is not to make you all afraid to go out or be in your soil, because resistance is a normal thing. And it's when it kind of tips to be too much that we're starting to see human health issues. And I do, you know, want to highlight that there are cases that antibiotic resistance can move from the soils and into crops. So that can be a way that we get exposed to it. Whether that results in antimicrobial resistant infections, you know, it's not like every time you eat antibiotics or antimicrobials, you're going to have a resistant infection. But I do think something that is really important to highlight is that there are certain people who are particularly vulnerable, who might need some extra care when it comes to antimicrobial resistance. And in particular, that's people who are constantly interacting with livestock. And so here, a study by Johns Hopkins saw that poultry workers have about 32 times more antibiotic resistance bacteria on them. And so that's a really important fact that we need to make sure we're maintaining healthy systems for people so that they're not getting higher rates of infections. And so, yes, it does kind of come into this awkward concept of farm to table where, we could be exposed to contaminated food or interacting with animals that are contaminated with resistance. And in general, though, I want to say about 20%, so 1 in 5 of resistant infections are considered to be caused by germs from food and animals. So yes, it is an important way that we get infections, but it's not the only way, and in fact, not the main way. So I know that can end up being a little bit of doom and gloom, but before I end, I do just want to say that there are some ways that we can all take action to limit the inputs of antibiotic resistance and also limit your chances of microbial resistant infections. And a lot of them are really simple, like washing your food. This is a pretty simple thing you can do that will help eliminate some of your risks of exposure. It's also really important to think about whether or not antibacterial soap is necessary for you. Most people in day-to-day life don't need to do the antibacterial version of soap. Normal soap can be great. And the problem with antibacterial soap is that it goes into our waters and enters into our ecosystems and helps drive up resistance. So think about whether or not you really need that extra potent soap or if normal soap can do. Also want you to think about, really exploring where your food and in particular your meat comes from. And this is just something that we should all probably be better at and ask, figure out what is the antibiotic use. Now, there's been big trends to just do all antibiotic free. And I think there are, pros to that, but I also want to say that it's complicated and, never using antibiotics ever might mean that sick animals are never treated. So it's complicated, but you do need to do your homework and feel confident that the way you're getting your food and is kind of the best for you and also hopefully the best for our ecosystem. You can also take really important actions to dispose of your medicines and in particular your antibiotics responsibly. Try not to just pour them down the drain or down the toilet. Most hospitals can take they'll take medicine buybacks or they'll just allow you to drop them off. And that's a great way to keep them from entering into our ecosystems. And then the last thing I recommend is to engage with your policymakers and encourage them to use informed regulations, in particular ones that consider this One Health perspective to make sure that we're using antibiotics really in the best way possible. So I've talked a lot about, the wide effects that these can have on our soil environments and also the potential impacts on antibiotic resistance. But I do want to say that I still think there's a lot of great questions out there. It's not all doom and gloom. It is complicated, but I think that's where the really exciting work is to be done. And I'm excited to continue on exploring these concepts. And so with that, I thank you all so much for listening. It's been such a pleasure to talk to you all about antibiotics and the age of antibiotics. And I would be more than happy to take any questions, but I'll throw it over to Julie, I think.
Thank you so much, Jane. That was wonderful. I learned so much.
Good.
Leaf cutter, ants, dung beetles, nitrogen cycling, that checks all the boxes for me.
Great.
So I just want to remind people that we'll be sending everyone a follow-up email that will contain the link to the recording for tonight's webinar. And also to remind everyone that next month's webinar is going to be given by Alexis Docius of the Native Plant Trust, and she'll be talking about understanding and managing soils for ecological health. So I hope everybody can join us on March 7th for that, and you'll be getting an email with the registration link for that soon. That'll be up on our website, so you can register for next month's as well. So we've got some great questions in the Q&A already. If anyone has questions, please go ahead and type them in. We'll start plowing through them. So the first question is, this is a question that came in while you were looking at the Burley's funnels. Is there a method for extracting invertebrates without killing them?
Yes, you can You just need to contain them, right? They're gonna run away from that heat source, and you can, if you don't need to like keep them in one, if you don't need to count them, and you just want to look at them, you can do that same thing. You just need to make sure that instead of having an open cup, because they'll go into the cup and then they'll walk out of the cup, and then you just have caused tons of insects in your house. But you can just allow that same process. You just want to make sure you've sealed. Sometimes what we do is we attach a mason jar lid, the ones that don't actually have, the ones that have the removable parts to them, and you can put that at the end of your funnel and then just screw the mason jar on, and then the invertebrates will go into that mason jar, and then you can take it off and put the lid on, and you'll have lots of living invertebrates in there. So yes, same process. It's just a little bit harder to count them if they move around.
It is, and they do not sit still. I know that from experience.
But it's so fun, and I think it's fun at any age, but it is probably one of the greatest ways I can get younger students and scientists excited about soil is to go through that process and just show them that there is an insane amount of diversity right underneath our feet that we just often do not appreciate.
Cool. Thank you for that answer. That'll be an awesome project for me and my kids to get into. That's really neat.
And there's lots of resources online. You don't have to memorize what I just did. Just look up Berlese or Tulgren, or even in, insect extraction funnels. Then you'll be able to easily go through that process.
We love finding bugs out in the yard in my household. So that'll be a hit. Thank you.
Yeah.
So our next question coming in is, why do you think the water holding capacity is changed by antibiotics? And I'm assuming that they mean in soil.
Yeah. So I think a big reason for that is it's really helpful to have lots of active living microbes in the environment, because they actually contribute to the soil matrix, and they are pulling in and, holding water inside of them. And so what I think is happening is that we're knocking out a lot of active microbes. And so those connections are not there anymore. That structure, the ability to act kind of like a sponge, is decreased just because you don't have that important biomass, and you don't have growing microbes that need to pull in more water. So I think it's just that you don't have enough microbial growth, and therefore you're getting that decrease in ability for water to stick around. It just kind of goes through. Nothing is pulling it in, sucking it up, and it's just moving quickly through our water system or our soil matrix.
That makes a lot of sense. One of the things that I used to do at the Cary Institute was measure water holding capacity. So I was wondering about that too.
Yeah. It's a fun intro to soil. Everybody has to do it at some point.
It is, and it's a good example of just how much is going on in soil that is not well understood. So
absolutely.
Yeah. So the next question is, what resources do you recommend to help identify soil invertebrates?
Oh, there's lots of great ones. I'll be happy to send those along, and I'd be happy to put that in our email.
Oh, that would be great. Thank you.
There are lots of different resources. It depends a lot on where you are. I started doing a lot of my work down in the tropics, and while there are orders of insects that are the same, species are quite different. But things like cooperative extensions are generally a really great resource. So Cornell Cooperative Extension likely has some options. Some of the kind of Farmland Trust, there are other different groups that might have some great options. There's also great work. I know some individuals at Cornell who are they have a great entomology program, and so they likely have local New York State resources, and it's possible that the DEC also does. So I'll be happy to add some links, but know that looking at your environmental groups at the state level is a really great way to get a good handle on diversity in your ecosystems.
Yeah, the more resources we can put in those emails, everybody loves to engage with their local resources, especially when it's about topics they're very interested in. So thank you for that, Jane.
Yeah.
Our next question coming in, is there any correlation between antibiotics resistance in soils and antibiotic resistance in people?
That is the $1 million question that I really hope one day I will get to answer. It is not a direct one-to-one. There are some times where that seems to be the case, but I would say that in general, soil is kind of the environment we don't have a great connection to human health. We know that if you look at antibiotic resistance in a hospital, that can be a good indicator of the type of resistance that's going to move into humans, but that's mostly because the microbes that are present in the hospital likely came from humans. And so it's kind of this circular feedback. But it's really one of the big questions I think has to be answered. And so my goal is to kind of connect with people who are on the human resistance side and start to get to these maps. Certain countries have better information than others about the types of resistant infections. But I think, there's definitely a way that I'd like to take our US-based map and say, here's what we see in the local environments. How much does that predict what you're also seeing in humans? But unfortunately, sometimes we just don't have a lot of, funding available to take human infections and figure out what it is they're actually resistant to. It's kind of trial by error. It's like, I guess they're resistant to tetracycline because it didn't work. So there's not always a systematic way of documenting human infections. But one day, that is a really important question. So I hope to one day answer that.
It sounds like there's a lot of people working on it now. So hopefully we can make some progress on that. So the next question is, would the pill bug also break down the soil in the same manner as the earthworms?
Oh, such a great question. I don't know is the very short answer. I think that, there's something really special about earthworms. They have a long gut system. It takes a while for soil to move through. And oftentimes earthworms are consuming soil in the way that we think of soil, where pill bugs are kind of consuming the microbial growth that happens on like leaf litter or parts of the soil that isn't typically what we think of as that like kind of gritty soil material, but it's actually the leaves and materials. They also don't have as long of a gut system. But I actually have done quite a bit of work studying isopods, and I know that they die pretty quickly when they're raised in antibiotic-laden environments. They do not do well. So part of me feels like maybe not, because they are not great at handling those compounds, but it's certainly a study that we could look at and is probably something down the line. But my instinct is that earthworms are probably better just because of how long it takes and how much they have this unique fermentation process inside their guts.
Cool. Thank you. I love, it's like you kind of point out the answers that seem so logical once you think about the length of an earthworm versus the length of a pill bug. Like it's something we should be able to connect to so instantly, but just since your expertise on it, it's just really cool to hear that.
Yeah.
And the next question I really, really like, because this whole microbial thing just blows my mind. I'm a social scientist. I think more about what people think about. So this question comes possibly from the comic book world. If antibiotics make cows grow larger, why couldn't the same thing happen for humans?
Sure can. And I think there is some studies out there that say that might be the case. Now, that being said, there are very few humans that are getting a constant subtherapeutic dose of antibiotics. So that's really, it's not like you take your week-long course and all of a sudden you're going to grow 40% larger. But if we were on these kind of consistent doses, it's very possible that might be influencing kind of the growth of humans. So it's certainly not my expertise because I don't work in humans, but I know that people have looked at it and the whole connection between what are the long-term effects of, particularly when you have to take multiple antibiotic courses, or if you have to be on them for a long time, what are those long-term effects? And are there major trade-offs? Most of the time that's happening because you have an infection, so you probably need those compounds. But I definitely think that there is no reason why, there's no clear reason that just absolutely couldn't happen in humans. It's possible it could.
And we're all just waiting for the day when we start to see that.
So
So kind of a related question here about humans. Does the use of antibiotics have an impact on our gut biome? And so are probiotics the answer to renewal? And maybe just also explain what probiotics are if not everybody understands that.
Yeah, so probiotics are, there's a wide suite of them, but they are ways that you can actually take active living microbes and consume them or have them put inside of you that could potentially replace a disrupted microbiome. The first part of your question is yes, absolutely. Antibiotics definitely change our microbiomes when we take them. But the long-term effects are really variable. You know, we find that in general, you can recover your natural microbiome. So all of us have kind of a stable microbiome that, you know, we could go out to eat and have different types of food, but in general, throughout a week, we're going to still have a pretty similar microbiome. It'll fluctuate a little bit. And you could take an antibiotic course and that might change it, but it often kind of reverts back to itself. Now, that's not always the case, and sometimes you get what we call dysbiosis, and your microbiome changes so much so that it really starts to have some negative consequences. So there is this huge growing trend to get into probiotics. Now, the big unfortunate answer is that most probiotics are not a magic bullet. They often will say, you know, we have 1 billion cultures inside of this, and you're going to get all these new microbes, but they're normally just like 1 or 2 types of bacteria, when normally your microbiome has hundreds and thousands, if not millions of different types of bacteria. So the idea that you're going to just drink a smoothie that all of a sudden replaces all of your microbes is unfortunately not going to probably fix most of your problems. There are really well-shown studies where you can get microbiome implants, and maybe somewhat, touchy one is the fecal microbiome, where people who have persistent infections in their intestinal tracts can actually get a microbiome transplant from someone who is deemed a healthy microbiome, and that can really change the infections inside of their gut systems. So we know that this idea of introducing a whole new microbial community can be actually a way to recover a microbiome. But it's complicated because sometimes what's the right microbiome for you may not be the right microbiome for someone else. So it's not a cut and dry answer. I'd love to say that something over the counter could totally change and fix your microbiome forever. But doesn't seem to be, we're not quite there yet. But there are some pretty complicated procedures that have really established new microbiomes.
And I think you must be a mind reader, because as you were saying that, a question came in about fecal transplants. So
Yeah,
it's really interesting.
Yeah, it's a really fascinating world. And, you know, it's not exactly my area of expertise, but I really love exploring it. It has been a lifesaver for some people, but it is complicated because, you know, you're moving all of these microbes, and maybe some of those are bad, right? And you don't want to bring in a new infection while you're trying to treat an infection. So there are times when it works. You know, it's pretty complicated, but it definitely is one therapy that I think in the next 10 years, we're going to see microbiome-based therapies just continue to explode in their uses.
Really cool, really cool. So the next question we have come in just is more of a statement, and I'm going to hypothesize a question out of it.
Sure.
It says golf courses and antibiotics. So I'm guessing that they just mean the soil health at golf courses.
Oh, it's such a good question, because so there are environments, I focused on livestock, but there are lots of environments that use a lot of antibiotics that you may not think about, you know, and it makes sense, but things like orchards, they use a lot of antibiotic sprays because if you get infections in your fruit, right, then all of a sudden you have a failed fruit crop. So a lot of golf courses will use pesticides, but they can be antifungals or even sometimes antibacterials as a way to keep their fancy lawns very nice and clean, but they are highly managed ecosystems. And I'm guessing that's what they're emphasizing is, you know, these are these are soils that are getting a lot of inputs, a lot of care, and certainly a place that I want to do more studying. Now, that being said, not very many golf courses want people out there taking a second golf ball-sized hole in the middle of their nice greens and sampling that soil. But I definitely think that they might be a really interesting environment to explore resistance. And yeah, so totally great question or statement, but it's definitely something I've thought about. I need to get a little bit more in with the golf community.
Yeah, you never know what you're going to find below ground. So the next question takes us back to earthworms. Is there a correlation between the decrease in organic carbon content due to earthworms and the earthworm's ability to aid in reversing the effects of antibiotics in soil?
So I guess I don't know if I totally understand the full part of the question. Well, in general, I don't know of a correlation between those two things, but we do generally think that earthworms are healthy components of the soil. They help aerate the soil. They help kind of turn over. And so this is really preliminary research on the antimicrobial resistance side of things, but it's possible that I think they said that there's less organic material. I don't know if that's always the case. I think in general that earthworms can help increase organic material. They process soil and they help kind of bring oxygen to keep microbes happy. And so I do think that there'd be a relationship between how much carbon is present, how much antimicrobial resistance present, and also how many earthworms you have. So there's probably some correlations there. Whether or not they're all causal is definitely a question we still need to explore.
Well, thank you. And what's looking like the last question for the evening is coming in, I think is a great one. Is there some transfer when we eat meat from animals who are given antibiotics?
Yeah, that is a really great question. In general, it's not a huge threat. The most likely times that would happen is if you're eating raw meat, because most of our cooking process is going to kind of kill off the bacterial communities and potentially kind of eliminate resistance in those environments to some extent. And so, similar to, you have to be really careful when you're cleaning chicken because you don't want to get salmonella, right? Like it's a very similar process where it's in the handling of those, and less so that you're, taking a bite of a steak and having all of this active microbial resistance present there. So we know that it can happen, but it's a pretty low level. And I would have to double check, but in general, there's not a lot of indication that microbes are migrating into the muscle or the tissues that we're eating. They're more in the digestive systems or on them because we haven't cleaned our food correctly, but it's not, that resistance is not necessarily being incorporated into the tissues that we normally are eating. So I'd say in general, the threat there is pretty low.
Well, thank you. And that was the last question. We don't have any others in there. We'll just pause a couple seconds if people want to type anything in. But thank you again for doing this wonderful webinar. I've learned so much, and this was just really, really fascinating to dive into this world. So if you can send me a list of resources for looking at soil invertebrates, we can include that in our follow-up email. So everybody can expect to get that from us in the next day or so. And this webinar has been recorded, so that link will also be in the follow-up email, and it'll be posted on our website. So you can come back and watch it anytime you want.
Great. Well, it was such a pleasure, and thank you all. It's so many wonderful questions. It's so great to talk to people about these things. So thank you all. And I hope that if you have any more questions, you can feel free to reach out to me at the Cary Institute or through my website.
Thank you so much, Jane. Everybody have a good night.
Yeah,
Good night,
thank
everyone.
you. Thank you, Jane, and good night, everyone.